everyone, my name is Natalie McCracken and I lead marketing here at Pattern Ag. And with me today, I have Danny DeSlover, who is our Director of Genomics for Pattern Ag. So today I have him on because I want to talk a little bit about when we take those soil samples in the field, what happens once they're sent to our lab? How do we turn that soil into really high quality DNA extraction so that we can actually turn those into agronomic insights? So Danny, can you talk to us a little bit about what happens once that soil hits your lab?
Yeah, thanks, Natalie. I actually want to start before it hits the lab and talk about the importance of how we're doing the sampling, which I know we might have covered before, but really the whole process is engineered for traceability. So when we have our samplers go out into the field, they go out and they're doing, very close 10-acre grids, 20-acre grids, sometimes even more dense for our chemistry applications. All those samples get barcoded and are scanned into our lab app. So we have a lab informatics system here that's tracking all of our processes. So the first thing that's really important once we receive the soil is we accession it, we receive it in. So that's a lingo term that we use for when we receive something. And then the soil samples, to get a good representative of everything that's in that bag, one of our special sauces is that we have high throughput blender device. It mixes up that soil really, really well. And so that way, when we take an aliquot or a subsample of that, we know it's a good representation of everything that's going on in that soil bag and everything that's going on in that field. From there, the real work in my team begins. We take that good represented sample and we do our best to extract the DNA and get it as clean as possible and and represent all the different microbes that are in there. And the challenge with that is that each microbe has different properties. There's some that are tougher than others. We have insects in there, we have leaf tissue in there, we have fungus in there, and all those have different physical properties. So we have a extraction technique that we use to blast open all those cells and wash away all the other cell debris, but also all the inorganic soil particles and materials. So we get a very clean DNA going into our analytical assays or analytic assays. So the important thing there is that we get a good yield of DNA. So we're looking for yield, we're looking for quality, how many contaminants are there? We're looking at the fidelity of the DNA. How long is it? How big are those strands? And so we do a lot of QC and then we normalize it. We take the extracted amount, we make sure we have the same amount going into each assay so that each sample is equally represented. And we test and we test and we test again, making sure that each time we get the same answer when we have the same inputs. So one way that we test and verify is we have sort of these mock representative samples where we'll put in a whole bunch of different things across the tree of life in the microbial world and see how evenly do we count all those things that we know we put in there. We also take real specimens that are agronomic problems like corn rootworm, soybean cyst nematode, and we'll put a known amount of eggs into a naive soil without any of those pathogens in there at known quantities. And then we'll go send it through a process and look out the other end and see, hey, did I count 50 eggs of corn rootworm or did I see 100 eggs of soybean cyst nematode? And we make sure that that process is continually meeting the benchmarks that we've established for sensitivity and specificity.
So based on what you just told me, which is super interesting, Danny, what happens if you go through your process, the DNA gets extracted, you put it through the different tests that we do to sort of get the results that go to our growers and to our dealers, and one of the zones, for instance, comes back and it's like, we don't have any information on it, because it happens very rarely, but it does happen once in a while, right?
So among all the things that we try to extract, soils are the most difficult and most challenging. Not because it's literally dirt. So we have a lot of organic and inorganic things. There's also a lot of things in the soil that inhibit these molecular tests and we have to make sure we wash all of them out. But soil is also complex. It's varied. Depending on where you are, it's going to be different. And the chemical compositions of all the different components of that soil are going to be different. And so it's hard to always get a consistent output because we might have something in a field that is like, maybe it's been sitting underwater and been waterlogged. And then there's a lot of like anaerobic bacteria in there, and that's drowning out the signal from all the other things that we're actually interested in. That's an example of something can happen. Or if fresh manure was put down, and now we're getting a lot of things that are coming from these manure in addition to the new inhibitors from the manure, that can be an issue. So those are the types of things that, we do our best. We try to repeat it if we don't get anything that we expect. And sometimes we'll go up to 3 times where we test it again and see, are we consistently not getting an answer from this particular sample? And that's kind of the nature of the game. It's the same thing in the human diagnostic space. Sometimes the test comes back as we can't determine what the result is. And that's an unfortunate and disappointing event, but it's part of the process right now. And we're working every day to make it better and improve that as much as we can.
Thank you so much for joining me today, Danny. This was super interesting. It was very different than any of the other interviews that we've done so far. So thank you. Thank you to your team for all the hard work you put in behind the scenes. And thanks for joining today. Thank you so much.